Right, good morning everybody. How are you all doing today? I hope you're having a good morning and you slept very well yesterday. Okay, today I would like to talk about my pain research. Okay, so let's start with defining pain. So pain is an inflammatory response signaling by abnormal cell membrane, or there's something called the TRPV1. So is evoked by a noxious stimuli. It could be by injury. What else? What's that thing called again? It could also be bacterial response. It could also be by shock. Okay, so these three things evoke or trigger pain. Okay, and there are different types of pain. There's chronic pain and there's acute pain. Okay, so chronic pain is a pain lasting for a long period of time, while acute pain is a pain lasting for a short period of time. Okay, so what's causing pain? Let's talk about the pathological or pathophysiology of pain. What's causing pain? So pain, since it's an inflammatory response, so there are different immune cells that play a key role. They all come together. Immune cells, they come release their cytokines and chemokines, and also there's something called the substance P, the prostaglandin 2. So when all these come together and they are reacting with each other, there's abnormal cell signaling, modulation, and mediation. Then this produces something we call an inflammatory soup. Okay, that's how we feel pain. Okay, so that's the pathophysiology of pain. Okay, and remember, pain is triggered by something called the TRPV1. So it's a receptor in one of the neuronal membrane network. So like when this is evoked, we feel pain. Okay, so TRPV1 plays a key role, and this also involves the TRPV1 genes. So the TRPV1 genes code for the TRPV1 receptor. Okay, so my research is all about using transgenic rodents. We're going to have albino and wild type as usual. So we're going to be doing like so some of the rodents are going to carry the TRPV1. Some are going to have a double, some are going to have a knockout of the gene, and some are going to have a double knockout. Okay, so knockout of the gene means some part of the genes has been altered. Okay, maybe the cytosine or the adenine has been altered. Why the double knockout will have almost everything knocked out. So maybe the adenine, cytosine, and guanine. So leaving the thymine unaltered. Okay, so that's how it's going to look like. And also we're going to be coding the cells with antibodies of mongoose. You know, mongoose. There's this video I always see